It's all right.
Moving on.
Okay, coming wide. Full wide.
Looks like it flattens out a little bit here on this Danny.
All right, well, let's head south then along this contour line where there was some rock.
Heading south.
Is it possible to come down a little bit to about 3 meters?
Coming down
2.5?
all the time.
Okay.
Yeah, I'm do we have a name for this computer? Yeah, Herc computer. Herc's coming down. I'm stepping down a bit. We're looking downhill now, so I'm just slowly dialing it in. And it's being slow to respond.
Your steps are 0.1. That might have
We're
something to do with
seeing
it.
some rockfish here, but the silvery fish that are swimming by are hake. Zoom on that rockfish below the lasers. Oh, it's coming towards us. That's a nice side view, very obliging. Okay, zoom out.
All right.
So it looks like we have some returning viewers, and they were wanting us to catch them up on any neat observations or discoveries since we towed to this new location.
Well, we're seeing a lot more rockfish and flatfish since we're in a different habitat than we were last night. We are on some cobbly, soft mixed sediment, which has some rocks nearby, so we're seeing some different species of fish. There's a hake there on the left. I'm seeing some rockfish. There's quite a few species of rockfish, so it's somewhat, it takes a little bit for some of us to identify them, including myself. But we're hoping Tom Leidig will be able to be on SciChat soon. He's a fantastic rockfish ID scientist.
It sounds like our previous watch found some octopus.
Ooh, I haven't heard.
Specifically the flapjack.
Oh.
Yeah.
Oh, that's why you said that.
Yeah.
Oh.
That was the first thing my science communication fellow prior to me told me. That's one animal I'd love to see on this expedition.
Those there's a couple different cucumbers here, but the ones with the white, you see some white frilly stuff sticking out of the sediment. Those are a burrowing cucumber, and they're just burrowed in the sediment, and those appendages are out catching plankton, and they bring those appendages in with all the food.
Let's take a look at that rockfish that's near the lasers, and there's one just below it, below the cucumber.
Copy that. You ready for a zoom?
Yeah, I'll just let go of the stick here and see if Herc can hold it.
All right, let's see what we can do. Okay. Okay, coming wide.
We can look at that guy since we're right there.
All right, let's zoom in.
Okay.
All right, coming wide.
One of our viewers is interested in some of the physical conditions of this area. What are the currents and tide like in the Greater Farallones?
Well, tides. We've got tides all around the world. Not, you know, I guess, what's the tidal range? I'm just thinking maybe 6 to 10 feet, maybe? Thinking high tide, 6 feet, low tides, negative when we have new moons. So a range there. And we've surely have different types of currents. We have surface currents and at depth currents.
Okay.
California current runs north to south. We have Davidson current, which runs
where they wanna
south to
go.
north seasonally. And with the seafloor features varied on the continental shelf and the slope, and having the Farallon Islands and the banks, there's definitely, in the canyons, we get different currents moving around those seafloor features, which influence the food at the surface. It's a key part of that surface productivity is how the currents move around those interesting seafloor features.
Thank you for that background.
I think, you know, as you get closer to the Golden Gate, we have more influence from the outflow of San Francisco Bay. A good portion of California's watershed comes out the Golden Gate and goes back in the Golden Gate, tidally influenced, and there is definitely a strong outgoing current out the Golden Gate. And in the Golden Gate on those major tidal changes.
Interesting. Is that why we saw so much megafauna when we were leaving the Golden Gate and San Francisco area? I think I saw a few whales. I saw seals.
I'm not so sure if it's a tidal. Oh, go ahead, Danny.
So we're seem to be dropping down slope.
Yeah, it is dropping off a bit as we head towards 2D.
Okay, I had wanted to stay along that contour line if it's possible to do that, just because that was some rocky habitat there. If we can stay there until we get there.
That would take us to the northeast up the hill, so
then we couldn't drop down to 2D from there.
we'd have to go east, then south, so we can do that, yeah.
In terms of San Francisco Bay and that outer area that we went through, I think there's a fair amount of mixing there of salt water, fresh water, because of San Francisco Bay coming out that way.
Sure.
I think the whales are following the food, wherever the food is, and we've had a lot of nearshore bait fish this year, and not so sure how much that tidal influence contributes to that, but they've been around, and sadly that's kind of in the shipping lane, so that's kind of a dangerous place for them to be.
Mm-hmm.
I wish we could just tell them to stay away.
Yeah, I've seen that firsthand in New York City. Our humpback population is really growing in New York City, but we also have a huge shipping industry, so although we see like over 40 unique individuals of humpbacks out there, unfortunately, there have been instances of them coming in contact with some of the barges.
Yes, we definitely have that problem here. Something that the sanctuaries are working on, trying to slow ships down.
Sure. Just getting all stakeholders at the table to understand needs in these conflicts of use.
Can we zoom on that sponge? The yellow
Yep, can do.
bumpy thing. Nav, it looks like I've lost
Alright.
my web high pack.
Will you give it a reload?
I have been reloading it. And I've tried taking off the index part of it.
Yeah. All right, stand by.
That kind of looks like a sunflower star.
Okay, coming wide.
Doesn't have that big central.
Yeah, we have a couple species of these big sea stars, the Pycnopodia and the Rathbanaster, and they look kind of similar. Rathbanaster is usually deep, but sometimes their range overlaps. So I thought we had been seeing Rathbanaster, but we might be seeing Pycnopodia.
Which would be great because they got hit hard with the sea star wasting disease near shore.
Can you tell us more about that?
Okay. I can stay here.
A couple years ago, our coast had an unprecedented event where sea stars started dying off and melting. It was really sad to see. And they called it a sea star wasting event, where we just lost a lot of these predators, especially in the nearshore and intertidal area. And a lot of factors could be involved in explaining why that happened.
Sorry to jump in. It looks like there's a mark across the seabed there. Is that my imagination? I'll turn and look for you.
Great, I've got it back. What's my background layer there? That pinkish, orangish.
Yeah, that's probably just the base bathy. Would you like something else?
I'd like the habitat interpretation, if you have it. There, okay. And the contours?
Yeah, this is not the same.
I missed that mark on the seafloor. Did we get an image of that, or did we not see it?
I did, yeah. It was just a
Okay.
small mark on the. This looked like it something bumped
Okay.
across the.
Yeah, and I
top
took an
of the
image
hill
of
there.
that.
Great. Okay, so this is a good some good habitat to be looking in, and we can follow this contour line over towards 2D.
Yeah, so I'm facing east right now. So if we head south towards 2D, that's down the hill.
I guess follow the contour that we're on now in the direction parallel to 2D. Does that make sense? I'm maybe not explaining myself well.
Yeah, but if you look at the contour map, the shades of gray are just a few pixels there. So to the south towards 2D or southeast is going to be downhill a little. I'll push on to the east here and see if the contour lines match up with what we're seeing. There's a little divot off to my starboard side to the south there. But maybe it flattens out here. It looks like maybe it does in the sonar.
Sorry, Jenny, just because I hadn't heard of the sea star wasting syndrome.
Oh, yeah.
Yeah.
So 2013-14, we had this event on the coast where sea stars just were dying. They're just sort of melting, limbs falling apart, and it's really sad to see, primarily in the near shore, but it was seen in the benthic area too, and it was all up and down the coast from Washington to, I think, all the way down to Mexico, even Baja. And to this day, they don't have a direct answer of exactly what happened. There have been events in the past, 70s, 80s, and 90s, but this one was the largest event ever in terms of the geographic area, the one that we had in 2013 and 14. And the sunflower stars, which are a star that have a lot of different arms, the ones we're seeing here, I'm not entirely sure if they're the Rathbunester or the Pycnopodia, the sunflower star. But they got hit pretty hard, and they haven't come back. And that's a concern for the ecology of the area. However, some species are starting to come back. They're seeing a lot of young recruitments in the intertidal zones. And there was some thought there was some linkage to some warm water, but I've been trying to keep up on this because it's such a mystery, and I just was checking online here, and to date we don't have a direct cause. There was thought of a viral pathogen, but that's unlikely in any species other than the Pycnopodia.
Yeah.
Which is that sunflower star. There's a group that's been at a UC Santa Cruz that has been tracking it and working with colleagues up and down the coast to track where they're seeing sea star wasting events and sea star wasting, and trying to get a handle of as much as they can. And if people see it, they can go to seastarwasting.org to contribute their sightings. But a lot of the folks that are doing intertidal research are well engaged and involved with that study.
And sea stars typically reproduce through external fertilization, releasing gametes into the water column.
Zoom on that rockfish, please.
Is that correct, Jenny?
I think so, yeah.
Zooming in.
Looks like
They
it might be a
release
green
eggs.
spot.
Sea stars reproduce, actually, they do this by releasing sperm and eggs into the water, and then hopefully they meet and settle on the ocean floor.
I suppose just that reproduction, form of reproduction can help populations to rebound faster than
Okay, we
other
got that. We can zoom out.
All right, zooming out.
So evolution is maybe on their side.
Well, hopefully good ocean conditions to stimulate reproduction is on their side. I know that a lot of animals reproduce in the springtime when we have that upwelling because so much food is in the water.
Zoom on that flatfish.
I can try.
Oh, we just got bumped.
Yeah.
All right, we can zoom out. There we go.
Okay, zooming out.
So that's what it looks like to the southeast, Danny. It's looks like it goes downhill a little bit there.
Okay. Okay.
So the local contour lines don't quite run to what you see on the nav screen there.
Okay, so we have gotten pretty close to 2D and been exploring this area for about an hour, which is what we had wanted to do. So if I could, we could talk about what we're going to do next, and you guys can tell me best way we can do this is we want to get from, let's see, we want to get from 2E to 2F, which I understand is about 2.8 kilometers. And then once we get to 2F, there's some more habitat that we want to explore and then do some transect. So could we run down to 2E and then cut across?
Absolutely.
It looks like we're going to get to some soft, flat habitat. So once we get there, yeah. So
2 east to the west.
let's see. How do we want to do this?
Southeast. And then
I'm
almost
sorry.
due east.
Roger. So I'm going to keep going southeast.
Yeah.
Roger.
You toggle.
Thank you. I'm home again.
All right.
I haven't been seeing it. And as we're heading over this habitat, I would like to zoom in on fish species, these rockfish species, so we can try and figure out which ones are here.
Yeah, copy that.
All right, we're going to be doing 0.5, so you should have a little bit of time to look at stuff.
Roger.
Dan, I'll take your direction. So as soon as you see one you think you can hold, I'll zoom. Just let me know.
Yeah, Roger. We're going to be doing half a knot, so they may be kind of on the fly zooms. Are you all right with that, Danny, or are you actually going to stop and look?
I think on the fly zooms are okay, and we'll try and get stills of those too.
Yeah, so you can, if you see one, Danny, you can shout out and you're free to zoom. Wayne, I'm just going to try and maintain a semi-constant speed here. Normally I would stretch it out tight and then stop and look. No logistics tonight, it's not desirable.
It's looking healthier.
It is.
Yeah.
Yeah.
I think she's actually side 2.
Can we veer off to the, well, he's swimming away. The fish to the left. That's okay.
I think that's a flag rockfish. Four vertical red and orange bands.
Okay.
Mm-hmm.
Okay, coming wide.
Slowly getting started.
There's a rockfish just to the left of the lasers. We can zoom on.
That guy,
Yeah.
the spotted guy.
Mm-hmm.
Stand
Oh,
by.
it's a little fuzzy.
That's a full zoom.
Okay.
That's as much focus as I can get.
All right, we can zoom out. So the little fish we're seeing are poachers.
That sure looks like a
wide.
sunflower star. Pycnopodia, the one on the right. What do you think?
0.5 knot.
Yeah, it could be.
Oh, 125.
Ooh. Yeah, it's a predation event.
Awesome.
We just missed it. It was just off camera.
He got away.
He's swimming away.
Oh, I see it there.
Hagfish?
So normally during this kind of mode I would stretch it to 100.
That's an interesting one. Oh,
Yeah.
little hunting going on.
I can't get as far ahead.
Can slow down too.
What's that?
Can slow down too.
Ship?
Yeah.
What was the predation event? I missed that.
I missed it too.
I believe one of the larger fishes were playing with one of the smaller fishes.
Oh.
Was not successful.
But it was an attempt.
We just saw the silver flash in the camera in the bottom lower right.
He was trying to make friends. Or dinner.
Yeah, it's jumped up.
Dan, are you okay with like a half zoom so we can get a little bit more detail on the bottom?
Let me come down a bit since we're going downhill.
All right. Here's the fish. Huh. Coming wide. He went.
That's a hake. We zoom on that rockfish by the lasers.
I can try. Ooh. There's a flatfish there too.
Yeah, nice. Okay.
Yeah, I think we'll see a little bit more if we're able to stay zoomed in. Okay. Yeah. Well, the straighter down you point, the faster we're going, the less time we have to respond. So they're like that guy. Ooh. See, yeah.
Yep. About
They'll be
2.5 meters seems to be pretty good.
okay.
But if we're zoomed out a little bit,
Yeah.
and yeah,
Let's get rid of the porch. Yeah, yeah. That feels all right. What? Yeah, understood.
that looks good for me. That looks good for you, Wayne.
Yeah, absolutely.
Okay.
Much better. Thanks, Dan.
0.3. It's better than 0.7.
There's another flatfish, I think. Just out of frame now.
Autos. No.
I know this is true of flatfish in New York waters, but most flatfish, they start off as symmetrical fish or having eyes on either side of their heads, and then their eye drifts as they mature and become this bottom flatfish. Is that true of the flatfish?
So we're at a really nice nice speed and a really nice height, and now we're in kind of this mixed habitat. So I'd like to maybe take advantage of this and do one of our quantitative transects in mixed habitat. So if we could keep this nice speed, height, and heading and do a 10-minute run on this and see if that habitat holds. So we'll do no zooms.
No zooms. No zooms. Understood.
And I'll go ahead and call that mark, and we'll get that in the data logger, and I'll get it in the notebook. And probably right when I do that, it's going to change habitat to mix to soft habitat. But let's give it a try.
Yeah.
Okay. So does that work for you, Dan?
Yes, sorry, I was off SBO. Right when you do that, I'm going to slow down just for a minute to let the ship catch up. We're playing with the speeds here. But yeah, that was a nice speed. We were doing about 0.5, 0.6 speed over ground. So we'll get the ship moving again, and I'll get it dialed in, and I'll give you a shout back when we're static at 0.5, 0.6.
Okay.
Carrie, to answer your question, yes. Same thing for flatfish over here.
Cool.
Can zoom on that rockfish while we're waiting, right?
Yeah, okay, with the scientist to zoom?
Yeah, because we're not on Transect, so yeah.
Good.
We haven't actually started yet.
There's two there.
Yes, there are.
Yeah.
Let's get some focus. There we go. There they are.
So we had everything dialed in there, and then we got out of the box.
Yeah, exactly.
No, I mean for the speed to get the whole show moving. Nautilus, Argus.
We got another one joining the show.
Yeah.
We have over 700 meters to cover, so we will find 10 minutes of constant speed.
That we will.
Okay.
Okay, while we're sitting, I'm going to make some notes.
Okay.
I've seen a couple krill swim by. Phausids. There's one right there with a little bit of red.
Oh, right. This guy wants his 15 minutes. There's another one. Is anybody else noticing increase in the hake when we stop? I think they're attracted to the lights, maybe?
I think they are too. They seem pretty curious.
Yeah.
It's good hake habitat.
Yeah, definitely when we stop, their numbers increase.
Looks like there's hake between Argus and Hercules too.
We just chased off the star of our show. What's this for?
Hake are also called Pacific whiting.
I think is he gonna bite his tail? Nope.
It's nice to have some fish around. Tell us which way the current's going. What's that?
We're going now.
Roger. Okay, here we go.
Alright, so this is full wide, so I'll just get rid of the equipment in the shelf.
Perfect.
That's where we were before for a zoom level.
Do you want me to drop a target at the beginning of this transect?
Yeah, that would be great. So we're all set to go?
Well, I just started moving, so let's give it a sec there, see how it works out. She, what's her speed over ground now?
0.6.
0.6. Hey.
Oh, we've got something up ahead. Oh,
We have
escape.
Something big. A ray? Or
Well, that skate there just to the left of lasers.
yeah.
Long, long nose.
Okay, it looks like we got the whole show all moving along nicely at half an hour, 0.6 to be precise.
Okay, this is looking good. Looks like we moved into soft habitat, but let's give it a try. Let's do a 10-minute run and see how we do.
Sure.
Another.
Oh, maybe not.
If you find skate eggs on the beach, you can.
All right, if we're all set, I'll go ahead and call that at
All set here.
12:44:30.
Okay. What about the skate eggs?
Oh, if you find skate eggs on the beach, we recommend that you do return them to the water because they can survive for a fair amount of time out of water. So there still could be hope for that skate, future skate. So skate eggs are often called a common name mermaid purses. And their eggs, yes, are pretty resilient. So pick them up, put them back into the water when you see them along the beach.
Yeah, a lot of these fish you can't see till they move.
So Danny, can you explain a bit of the purpose behind running a transect and what a transect is exactly?
Sure. So a transect is, we just have a designated line. We either pick start and end points, or we have a standard bearing and go by time. So this one we're doing by time because we're just designating it on the fly. The point of doing it is that we stay at a constant height, speed, and either on the transect or at a heading, and it keeps all those variables the same so that when you count organisms along during that time frame, that all those are constraints are the same, so that you're always looking at the same amount of area, and you can look at the number of organisms in that area and look at the density of organisms. So it's a way of making sure that you can quantitatively assess this, or statistically assess the animals in that area. So it's a little just more standardized.
Great.
There's a boot sponge or a barrel sponge.
And that's to help determine or estimate the population? Or
Yeah, it gives it helps to quantify the organisms in that area. And then if you wanted to repeat these surveys later, so for example, in these areas where the fishing management is changing, if you wanted to assess how those changes had affected the habitat, the communities over time, if we wanted to come back in a few years after there'd been some fishing in this area, and then we could run these transects again, and we could say, we could then count the organisms in that same area and see how it's changed. So it gives you a standardized methodology to use.
thanks.
And in science, we always work to having this standard method, so then you can replicate it, as Danny was saying.
these hake are coming along with us.
Our guides.
Yeah, our
At-
groupies.
Yeah, 305 meters.
That big fish over off to the right, the one that's trying to be a hake, that's sablefish, also called black cod.
Okay.
We now have an escort.